On behalf of the Patient Safety Authority, I would like to welcome you to this webinar titled Instrument Cleaning, Sterilization, and Storage for Infection Preventionists in Ambulatory Surgery Facilities. My name is Joanne Atkins, and I will be your moderator for this program. We welcome any comments or questions, and all materials are provided for your educational use. Now I'd like to introduce our speaker for the webinar. Denise Cutting is a registered nurse and infection prevention advisor for the Patient Safety Authority. In this role, she serves as a resource for the Pennsylvania facilities through education and the development of infection prevention initiatives. Denise's clinical experience includes med-surg, telemetry, and performance improvement. Prior to joining the Patient Safety Authority, she was the regional manager of infection prevention and control for the Geisinger and Penn State Health Systems. None of the planners for this educational activity have relevant financial relationships to disclose with ineligible companies whose primary business is producing, marketing, selling, reselling, or distributing healthcare products used by or on patients. Denise, I will now turn the program over to you.
Thank you so much, Joanne. And thanks everybody for joining today. Nice to see some of you attending who I recognize from our symposia that we had. It's great that some of you were able to join today. So we're talking about things related to sterilization, high-level disinfection for ambulatory surgery centers, but what we're talking about today would apply to other facilities as well, not just ambulatory surgery centers, but the purpose of this webinar and the reason we're doing this today is because of the conversations we had with facilities during our Keystone ambulatory surgery presentations that we had. So thank you all for being here. Hope you're all staying warm out there today. And please do, as we go along, any questions you have, please do put them in the chat. I will try to address them as we go through. And if we don't address them as we go along, we'll get to them at the end. So our objectives today are to describe the basic knowledge and skills for sterile processing. Now we know that there is so much that goes into sterile processing that we're not going to be able to get into all of the details today, but we're gonna cover some basic knowledge. We're going to talk about decontamination, cleaning, disinfection, sterilization, kind of going over what the difference is between the two of those. And finally, we'll talk about water quality because it is really, really important that the water that we're using to clean our instruments is what it should be. So when we talk about sterile processing, there's a lot of different terms that can be used interchangeably. One of those is point of use. And this is really when we're talking about instrument decontamination right when an instrument is used, usually in the procedure room or in the operating room. And we'll talk in a little more detail about this later. But we know when we're doing point of use cleaning, we need to use appropriate PPE. And we'll get into more of that a little bit later on as well. Decontamination or cleaning can be used interchangeably again, but when we're talking about a decontamination area, it is a specific place where we're removing gross bioburden from an instrument. And we'll get into that a little more later as well. When we talk about water, there's utility water and critical water, and of course, tap water, distilled water, sterile water. There's all kinds of water out there. When it comes to sterilization and high-level disinfection, really we are talking about critical water most of the time. That's usually what we want to use because that water is extensively treated to remove microorganisms. In some cases, utility water can be used for flushing, washing, and rinsing. Sterile instruments, when they are are ready to be packaged. So there's all different types of packaging that can be used on sterile instruments, containers, ways to identify sterile instruments. We'll get into that. Sterilization itself is a process by which you're removing microbial life from an instrument. And it includes all microorganisms, bacteria, viruses, spores, all of it. But when we're talking about high-level disinfection, there may still be some mycobacterium type species left behind. So basically the spores sometimes are potentially still left behind when we're talking about high-level disinfection. So anytime we're looking at an instrument and how we're going to clean it, we really need to follow those instructions for use for that instrument. So manufacturers put out and test certain chemicals on their devices to determine what is effective at removing microorganisms from that device. So that is why we have those instructions and why we need to make sure we're using them to the best of our ability when cleaning a device. So we need to use the right chemical, the right dilution, depending on what it is, and the right time and coverage. So whenever manufacturers are testing the effectiveness of chemicals at removing microorganisms, they look at different times, and that's why they determine what it is that they recommend. So it's really important that we follow those instructions and follow the right time and coverage. So sometimes in my experience, I've seen instruments or devices where you'll have a product that's recommended for use to clean that device. And sometimes the dwell time or wet contact time, depending on what you call it, that would be recommended by the manufacturer might be longer than the duration of time that is recommended on the product you're actually using it in itself. So an example would be a bleach wipe. So generally bleach wipes, you would use 3 or 4 minute dwell time, and that's usually what the manufacturer of the bleach wipe would recommend. But I've seen situations where there's a product or a device that has been tested by the manufacturer of that device, and they may recommend a longer contact time, wet time, dwell time for that. So really important to make sure you understand what the manufacturer is saying to use for the device. So again, sometimes we use sterilization and disinfection interchangeably, but they aren't exactly the same. So as I said earlier, sterilization is considered more effective because it does kill spores and vegetative cells. Disinfection reduces germ count and only kills vegetative cells, not always spores. So when we're talking about sterilization, it can be physical, manual type removal of microorganisms, chemical. We use heat, radiation, and filtration. And a lot of times with disinfection, it is primarily just chemical. But both are effective at reducing overall microorganisms on a device or piece of equipment. So we're going to go into our first polling question today. So at your facility, I'm curious to hear from you, what type of high-level disinfectants do you use? So we have glutaraldehyde, OPA, hydrogen peroxide, peracetic acid. It's all right if you don't know, but if you could take a minute and you can select more than one, maybe you use more than one product at your facility, but take a minute and let's see what kind of products you guys use out there for high-level disinfection. And if you don't do any high-level disinfection, that's okay as well. All right, Karen, so let's take a look and see what. All right, so 42% of you use hydrogen peroxide, Not surprised by that. Next, 32% use OPA. About 30% of you aren't sure, which is okay. And maybe you'll, go back and see what kind of products you're using after today. Glutaraldehyde, 26%, and peracetic acid is 22%. So we're going to go over a little bit about these. We're not going to get into tons of detail, and there are others, of course, out there that you can use. So these disinfectants are regulated by the FDA, and some require different combinations or mixing. And there's a minimum effective concentration for each of these, so that it, again, kind of when we talk about disinfection, as I did earlier, you want to make sure the concentration is what it's supposed to be based on the manufacturer and your instructions for use to effectively kill the organisms that are needed to be killed. And it's important to know the safety features and PPE that's needed for these different types of disinfectants. Glutaraldehyde, another name for it, Sidex. Probably the least liked high-level disinfectant, I would say, maybe, because it does have a strong odor. It must be mixed, and it is used for heat-sensitive equipment. But there have been some reports that it can cause some adverse health effects for staff. And, the fact that you need to avoid skin contact, and it needs to be used in well-ventilated areas. So some facilities are moving away from using this product to other things, if they're able to, based on the equipment that they have. OPA is generally normal heat or steam sterilization, does not require exposure monitoring. Sidex does, but OPA can stain. So again, you want to make sure you use appropriate PPE when using that. Hydrogen peroxide, probably the one that most people are familiar with. You can have it in your house. So, it is readily available and out there. And what it does is it destroys hydroxy-free radicals, and it attacks the membranes, DNA, and other cell components. And if you use it at home, right, that bubbling, it's kind of attacking cells. And then peracetic acid is good at removing organic material because it's acidic, but it can corrode instruments. So, each one of these high-level disinfectants has risks and benefits with it, but it depends on your facility and what instruments you have and devices that manufacturers might recommend using each one of these. But really important to follow PPE recommendations and make sure you're being safe when using these high-level disinfectants. So thank you. Thanks for participating in that polling question and going over those. So Spaulding classification, if you've heard of this before, maybe if you're studying for your CIC exam, this is definitely something you would want to know about. So this is how you classify different types of devices or instruments, and then the type of cleaning that is required for them. So critical devices, objects that enter sterile tissue or the vascular system and require sterilization. So the highest level of disinfection would be sterilization. So surgical instruments, implants, needles, anything that's really entering into the body. Next would be semi-critical. So these are objects that come in contact with non-intact skin or mucous membranes. So this is where endoscopes, this is why we use high-level disinfection with endoscopes and probes, anesthesia equipment, and some other things. So these are just some examples. There's definitely more out there of what would fall into each of these categories, but these are just a couple examples. And then non-critical, these are items that come in contact with intact skin. So we're talking about low-level disinfection. So blood pressure cuffs, thermometers, that sort of thing. So generally, when we talk about low-level disinfection, we're talking about the different types of wipes you use at your facility to clean different devices. And as we're going along, so please do share. I do see there's some questions in the comments, so we'll get to those as we go along. And those are some links that it looks like Adrian Bell shared for us. So thank you. So when do we need to clean instruments? Obviously, anytime we use them during surgery or a procedure, even if they're instruments that are used in a case, they're in the room for a case, but they're not used, they would also still need to be processed after that, especially, I mean, if they're in an area with a patient, then they would need to be cleaned after that. Any instruments that come into your facility need to be sterilized if they're going to be used on patients. If they're out for repair, stored as backup. So sometimes we have instruments that might be stored for a long period of time, and they may need to be processed again if needed. Obviously, if things get contaminated, if you have any problems with temperature and humidity, depending on how long that may occur, that can influence your instruments and whether they would need to be processed again. And again, any type of loaner instruments. Now, I know that this is sometimes a struggle, depending on where you are, and you have reps or people wanting to use things and they want to bring them into your facility and they want them processed really quickly. So, you know, I know that that can be a struggle, but they need to be processed thoroughly prior to use. You know, when I think about this, I think about, you know, what I would want if I were having surgery or my family member. You know, I would want to make sure that where I'm having a procedure or a surgery that they would want to do everything to make sure that the instruments are as clean as possible, because I know I wouldn't want to have surgery for anything like that. So a question, how often do backup instruments need to be reprocessed? Well, I wish I could give you a straight answer on that, but it depends. It depends on what instrument it is. You know, some of your instruments or packs will have dates on them, or you have the tape, the indicator tape that would tell you whether something needs to be reprocessed. So it depends on what it is, potentially. So when it comes to outside of temperature and humidity, you should have a policy based on that. So different places use different standards, and generally, if it goes high humidity is where you would be more concerned than if it's low humidity, because low humidity isn't going to lead to more bacterial growth generally, but that can lead to other concerns. So when things go outside of range, it really depends on your policy, and you would want to work with your facilities and obviously infection prevention leadership to kind of discuss what your process would be, depending on the situation and the time frame and which direction you're going, temperature and humidity wise. So sterile processing department, obviously you would want that area to be as centrally located as possible. Now I know in a lot of situations that may not be the case, but you ideally want your sterile processing to be centrally located, easy to get the instruments to safely. And you need to be monitoring your sterile processing area. Again, dirty to clean. Soiled, you really have to have separate spaces the best that you can. I know that this is difficult in some areas, but you really need separate spaces. So a soiled area, cleaning and processing area, and then your sterile storage area. So your storage and all of these areas need to be monitored for pressure, and especially your storage area for temperature. And your storage areas, I know that these are challenging spaces sometimes too. Ideally, you would not have windows in your storage area. I've seen this places because it affects your temperature and humidity when you have windows. Those areas, you shouldn't have exposed water, sewer, air conditioning lines in those areas. No exposed light fixtures. You want to check for moisture, leaks, that kind of thing. Positive airflow out of the area for where you're storing sterile instruments. And ideally, you would want to keep your door closed in your soiled area because you really need to make sure air is flowing in the right direction. We don't want to have dirty air flowing into clean spaces and vice versa. Okay, so we're going to talk a little bit more detail about point of use cleaning. So this is relatively new, I guess, over the last several years. It became a standard for the Joint Commission and some other places. But basically, anytime an instrument is used, at that point of use, the cleaning process begins. In the operating room, in the procedure area, that's when the cleaning begins. So ideally, you would, rinse instruments to remove gross bioburden, using the right kind of solution to rinse that instrument because we don't want to cause damage to the instrument. So depending on what it is, that may vary. The point of doing this is to make sure that biofilm to try to stop the process of biofilm forming on an instrument. Especially in the crevices, creases of the instruments, following the instructions for use as well. So some instruments, there are different types of wipes and rinses you can use depending on your facility. Some of them are, not really advocating for any of them in particular, but basically gross bioburden needs to be removed in whatever fashion works for you, following the manufacturer's instructions. And then once the gross bioburden is removed, then the instruments really need to remain wet, using pre-cleaning solutions. Again, there's lots of different ones out there. And depending on your situation and when your instrument can be processed by sterile processing, you'll need to know that the solution you're using would keep the instrument wet for a period of time if need be. So an example would be, if it's a case at the end of the day and you're not going to be processing the instruments till the next morning for some reason, or if that's your process at your facility, you want to make sure whatever solution you're using is effective for whatever period of time that you need. If it's 12 hours, 18, whatever it is, 8 hours, 12 hours, they will say how long they're effective at keeping instruments moist to stop biofilm from forming. Ideally, you want the instruments open and disassembling to a certain extent, depending on the instrument. Single use is obviously best, but we know that that's not always the case. And then your instruments need to be safely transported in hard containers to keep everybody safe. So we're going to get into a little bit more about how biofilm forms. kind of a way that I think about it, which hopefully none of you are eating right now, but you know, when you eat something, or say you have syrup on a plate and you just kind of sit it out and you don't soak it or rinse it, it makes it a whole lot harder to clean that plate. Whatever it is, you know, pasta sauce, or you're cooking pasta and it sticks to the bottom of the pan and you don't soak it, that makes it that much harder to clean it off. So a little bit different when we're talking about a biofilm, but same kind of idea. So basically, once something attaches to an instrument, then things are much more likely to grow, which makes it even more difficult to remove that biofilm for your sterile processing department to be able to do that. So really, it is really, really important, that point of use, to remove as much bioburden as possible. And we're talking about blood, dirt, sputum, body oils, urine, mucus, all of it can really just kind of help biofilm form and just make it really difficult to clean instruments. So in your decontamination area, engineering controls, like we talked about, humidity, negative pressure, air exchanges should be at least 10 per hour. Should have an eye wash station because of the, you know, the chemicals that we talked about, the high-level disinfectants. You know, you have to, some of those you have to have an eye wash station with them, located within 10 seconds or 30 meters of potential exposure. Have to have PPE, you know, full gown, eye protection, mask, face shield in some situations, really important. And obviously hand hygiene, which I haven't talked about yet, surprisingly, in an infection prevention presentation, but hand hygiene sinks separate. So you have your decontamination 3-sink system, and then you have to have a hand hygiene sink separate from that. So in the 3-sink arrangement, you have a wash, a rinse, and a final rinse. And then your water temperature, generally 80 to 110 degrees Fahrenheit, which sounds pretty nice right now, or 27 to 44 Celsius, you know, depending on the instructions for the manufacturer instructions for use. There's all kinds of different brushes, cloths, sponges, depending on the type of device that is being decontaminated. And lint-free cloths are generally needed, especially for endoscopes. So different types of PPE, generally standard or full PPE. I think full PPE is the best with a face shield because they're, you know, decontamination splashes are pretty common. And a face shield really is best. Enhanced PPE, not necessary generally for decontam. All right, so we're going to shift gears to different types of cleaning processes. So we have another polling question for you. So what type of cleaning processes do you use at your facility? So do you do manual cleaning? Again, you can select whatever applies. Do you have ultrasonic cleaners, washers, disinfectors, cart washers? If you're not sure, that's okay. Or if you don't use any of them, that's okay too. All right, I see a couple questions I'm going to try to address before they disappear from my screen. Recommendations for cleaning or disinfecting brushes. And it depends on the instructions for those brushes. Most of the time, some are single-use brushes. Some brushes can be disinfected and cleaned based on whatever their instructions are. Some say you can use them a certain number of times, so it really kind of depends. Again, I keep saying that, unfortunately, but it depends a lot of times on what the instructions are for that particular brush. And can your eye wash be over one of your decontam sinks? I don't think so. I'm not, your eye wash, I think, has to be a clean sink, not on a decontam sink. All right, so we can close up the polling question, Karen, please. Thank you. Okay, so 81% of you are doing manual cleaning. 80% have washers, disinfectors, which is great. Ultrasonic cleaners, 73%. Automated cart washers, 38%. And some of you aren't sure, which is okay too. And there are some other ways to clean items, but that you might use out there too. So not surprised that a lot of you are doing some manual cleaning, but you do have some automated things, which is great too. So ultrasonic cleaners, they remove soil, and they can really get into the crevices and lumens of different instruments, which is why they're really good. That they create the vibrations to get in and dislodge debris within the instruments. Sometimes high temperatures of these in these ultrasonic cleaners can kind of adhere proteins to the instruments. So again, just really important to make sure the point of use cleaning is done effectively, because sometimes if you don't clean an instrument effectively at the point of use and you put it in one of these cleaners, it will adhere that whatever it is to the instrument. So these are low foaming detergents that are used, and you don't want to mix different types of metal, I guess, if that's what I'm trying to say. So in these cleaners, if you have different types of metal, it can cause issues. I don't really understand the science on how that all works, but it can cause problems. Basically, it's a transfer of ions that can cause issues if you're using different types of metals. Washers and disinfectors. They use spray force action pressurized water against instruments to remove the bioburden. Different combinations of temperatures, detergents, and times, depending on the instrument and the type of washer or disinfector. Some, mineral buildup can happen in these, depending on the type of water source you have, which we'll talk a little bit about later. And then cart washers are like automated washers, but they have limited option for phase cycles when you compare them to a washer or disinfector. So obviously sometimes instruments need to be cleaned manually. Things with lumens, delicate devices, or if the manufacturer will say they have to be cleaned manually. Eye instruments tend to be one of those that are manually cleaned. And some facilities that don't have mechanical cleaners, your only option is to manually clean. So really important to be careful, obviously, when you're dealing with these. Safety is really important for your staff that are doing these manual cleanings. So different types of sterilizers. There's immediate use sterilizers, flash type sterilizers are out there. High temperature sterilization tabletop, gravity air, dynamic air, pre-vacuum steam, steam flush, and then low pressure sterilization. So there's lots of different types of sterilizers out there, depending on your facility and what your needs are. All different kinds of things that can be used to sterilize your equipment. So whatever we're doing, whether it's manual cleaning, whether we're using sterilizers, whatever it is, we need to make sure that we are adequately cleaning the devices, looking at quality indicators. So looking at cycle time, temperature, and pressure for different things, and recording You need daily logs to make sure that chemical indicators are being done, biological indicators. Usually we're using bacillus spores. And, you know, those things are used in the cycles to make sure that things are sterilized adequately. For steam sterilizers with pre-vacuum cycle, you have to do a Bowie-Dick test on those. And all of this needs to be documented. And in policy, you know, you need to have this in policy for your quality monitoring, as well as what happens if there's a failure. So what is your process if you have a sterilizer that fails or a chemical indicator fails? What's your process to deal with that at your facility? Should be outlined in policy. So once your instruments have been sterilized, decontaminated, you have your preparation and packaging area. Ideally, you have that separate and it's positive air pressure because you want that air flowing out. Rather than into the area, and it, obviously reduce the risks of airborne pathogens getting into that space. Temperature and humidity, which we talked about. These areas really need to be clean, really need to work with your environmental services to make sure that these areas are free of dust, because you don't want dust getting into instruments that are going to be packaged and possibly used on a patient. The workstations need to be cleaned at least every shift. And the other things that go along with, operating room, when we're talking about nail polish, hair coverings, restricting traffic, no food or beverages. So all the things that you would do in an OR, you want to do in your packaging area as well. So there's a question about one tray immediate use sterilization. So flash, kind of one tray is, I don't know if it's necessarily immediate use sterilization because it does go through the process. My understanding with one tray is it just doesn't have the drying time compared to some other processes. So generally, infection preventionists are not a huge fan of one tray, but the research is still kind of out there, I would say, based on things I've looked at, and obviously the representatives of those companies feel that their product is really effective. And some people that I've talked to have been using it effectively without any issues. So, but flash sterilization, ideally you want to avoid. And any of those, you need a policy on when you use it. So sometimes, one tray can be used if you have reps that are bringing in or surgeons that want a particular thing to be used because it's quicker, technically, some of the time. So, you'll do one tray, but ideally you want to be able to follow your full process to ensure that all the quality indicators are met. From an IP perspective, we generally you want to be able to follow your full process, but some facilities are able to use one tray pretty effectively. But ideally, you want to have your things ahead of time so that you're able to follow your process and really use one tray limited, and obviously flash sterilization very limited in emergency situations. So you use towel. Somebody commented that they use towels to dry after manual cleaning. Hopefully there are lint-free cloths and towels out there, or low lint. Ideally, you want something like that before wrapping. Yeah, I could see why you would want to do that. But you want to limit the amount of dust and lint as much as you possibly can. So also in your packaging area, you really need a good magnification system in place, good lighting so that instruments can be visually inspected. Because even, you know, you're going to go through your point of use cleaning, you're going to go through your sterilizer, you know, everything's going well. There could be situations where an instrument's damaged, that it may get missed until it gets to the inspection phase. There could still be bioburden on an instrument, even if it's gone through the whole process. So really important that instruments are inspected using magnification, looking closely to make sure before they're packaged that that everything is good with that instrument. That's a great question, Rachel. I don't know. I don't know if there are requirements for the power of magnification used. I've not heard of that, but we can find out. I don't know the answer to that question. That's a great question. So different types of sterile packaging are out there. They should be FDA approved, specific for what they're being used for. Rigid containers are needed for some things. Peel packs can be used, flat wrapping, and then labeling is important to make sure you know when things are, like as I had mentioned earlier, if you have dates, if you have a process for your time frames at your facility per policy, when something would need to be reprocessed, that kind of thing. So all those things are important when you're packaging your instruments. All right, so we're going to switch gears to endoscopes. So endoscopes are more difficult, I would say, than some, you know, different instruments, because it is a little bit more difficult to visualize, obviously, when you get to that end point, what's going on within the endoscope. And there's a high risk with endoscopes, and because of all of the steps that are needed. So, but it is very similar to other types of instruments. You still do the point of use cleaning, decontamination, but with endoscopes, there's an addition of leak testing, which you don't necessarily have with other instruments. Visual inspection, if possible. Ideally, you have a way to do that, but some facilities don't. Drying and storage, and then reassembling the scopes. So we're going to go through these a little bit. Failures in this process can really lead to poor outcomes for patients. So, you know, we still hear about outbreaks relatively commonly in different areas because of failures with the reprocessing of endoscopes. So this really, really is important. A lot of the times it's duodenoscopes for ERCPs. Those are the ones that tend to be the highest risk when it comes to processing effectively for patients. So it really is important that all of the steps in this process are followed as they're supposed to be per the manufacturer. So for scopes, you know, visual inspection would generally be with a borescope type camera, so that you could, once your scope has gone through its process, that you can look down in there to see if there's anything left behind. Leak testing, we're going to get into that a little bit more, and then ATP testing. So ATP testing is basically you are looking for any proteins that would be left on a scope from an organism. So basically any type of organism would generate these proteins, and that would, if you have a positive ATP test, then there's something going on with that scope. It has not been adequately cleaned. And now if it's gone through the process, it might need to be processed again, but if you continue to have an issue, then it might need to be something that would have to go back to a manufacturer to have a look. So any sort of quality issues with scopes, if you're following your process 100%, you're using timers, you know, you're doing all of the things, and there's still issues, then a lot of times you have to work with the manufacturer. So ideally, you would have disposable scopes, right? But we know that those are expensive and not generally used out there yet. But it really is the best thing, if possible, to use those. So for leak testing, again, you're following the instructions for use. Really, any type of leak can cause so many problems, and you would not want to use that scope on a patient because then there's clearly something going on there. So it's not adequately cleaned, and then you would not want to use it on a patient. There's different types of leak tests depending on the type of scope that you have. You want to use PPE anytime you're doing leak testing to keep your staff safe, and following the instructions for use anytime you're doing leak testing. And if you have a failure, just like other things we've talked about, what's your process? Do you retest it? And then if it's an issue and it continues to be a problem, then you may need to work with a manufacturer for that product. So manual cleaning of scopes may be necessary, and really important that you follow all of the steps adequately, as we said earlier. Adequate soak time using timers, cleaning and rinsing, removing all of the debris like we've talked about. Solution concentration is really important for endoscope processing. Verifying each time you use it that it's the right concentration and it's the right temperature, and minimizing the evaporation. So you want to try to keep those solutions covered if you can. And shelf life is important, making sure you're not using things past their expiration date. Really anywhere throughout the process on anything we've talked about, which I hadn't mentioned until now. So, making sure things aren't expired is also important, because again, that's what the manufacturer says, and has tested the product for its effectiveness. So that is why we need to follow expiration dates, because it may mean that the product is not effective if we use it past the expiration date. And then drying is really important for endoscopes as well. So you're rinsing with water, and you need a separate space for that. Critical water is required for rinsing for endoscopes, as we talked about earlier. So water that has been, all microorganisms have been removed. Leak testing, no visible moisture, using a lint-free cloth to dry the exterior. And then you want to not attach everything back on it right away if you can, so that it can dry adequately. And then there's different ways to store scopes. What I'm used to seeing is hanging racks, but where they're stored vertically. But I guess there are some storage things out there now that are horizontal, which, if that works for your facility, I think that's probably okay. And there's no set storage time for scopes, and you should do a risk assessment really for your facility to determine how long scopes are going to be stored before they would be reprocessed again. There's different times out there that I've seen at different facilities, but for you at your facility, you want to make sure you're following your policy and that you've done a risk assessment to determine when you're going to reprocess a scope if it's not been used for a period of time. Automated endoscope reprocessors, AERs, do still require manual cleaning, just like anything else, and leak testing. So, still like you would with other instruments, you're still going to do like your point of use cleaning, And then you're still going to look at quality indicators for that scope. But kind of like sterilizers, it is a more timed, controlled process than manual cleaning. And more consistent with the type of water, cycle parameters, that kind of thing, when you use an AER. So as I said, storage, clean, well-ventilated, secure locations, vertical or horizontal, and then the time frame depends on your facility. So in the resources, there's a good link to water quality in more detail. But water quality at any healthcare facility is really important for all aspects of care, not just processing instruments, but especially for processing instruments, because if you live, like I live in an area with really hard water, that will impact your sterilizers, everything at your facility, if you have different water quality issues. So water management team is really important. It can't just be the infection preventionists by themselves. You need a facility, you need leadership to make sure that everything is in place, that things are being tested appropriately, and that things are where they should be to keep your equipment functioning and your devices and everything functioning to the highest abilities. So it's really important that you have a group that is making sure the water quality is the way it should be to keep everything the way you would want it in your facility. Everything is really impacted by it. Sorry, I'm just pausing to look at a couple questions. So there's a question on how long do we need to keep documentation related to high-level disinfection and sterilizing, 3, 5, or 10 years? That's a good question, and I don't know the answer to that one either. So we will have to look at that, and we can circle back here in a couple minutes, Joanne, if you have answers to any of these questions. So there's just a couple more slides, and then at the end we do have a resources tab where some of these things are. So I'm documentation related to high-level disinfection. I want to say at least 7 years, based on some, but I don't know for sure. So we'll circle back on that question too. So the different things you want to look at for water quality: pH, bacteria, obviously, endotoxin, hardness, alkalinity. And these are just some examples of what you're looking for each type of water. So steam is also important. Obviously, you would test condensed steam to look at this, look at these different areas, and where things should be for your facility. And then how often they should be tested. So pH should be done quarterly for utility water or monthly. So, I don't know. For me, I would want to do a consistent process for both, but these things do have costs associated with them. So, whatever works best for you for your facility. But all of these things need to be looked at either quarterly or monthly, generally, for each of these different types of measurements when we're looking at water quality. All right, so I hope that I, as we went along, addressed some of the questions that I could, and those that I don't know, we will circle back on unless Joanne has any insight to some of these questions.
Thanks, Denise. This concludes the slide presentation portion of our program. And now, like you said, we'll do the question and answer period. We do have one question, which was, when should immediate use steam sterilization be used?
When we're talking about one tray, immediately use sterilization, you want to use it per your policy. So you should have some sort of outline as to when you would use it if you're using it at your facility. And ideally, you want to reserve it for certain situations and not rely on it for most of your sterilization, if that helps.
Yeah, thanks, Denise. And for the person who asked the question on whether one tray was immediate use, they are two separate entities. Immediate use sterilization can't be stored. It must be used at the time of sterilization, whereas one tray has been approved by the FDA for storage up to 48 hours only. So they're not exactly the same thing because you do have a little leeway with one tray to store them for 48 hours.
Thanks, Joanne.
Yeah. The question that was asked on how long do you need to keep documentation related to scope, high-level disinfection, and sterilization. As Denise said, that one's kind of sticky. Many standards, such as Joint Commission, say 5 years. However, I would also check with my legal department because we know that in the state of Pennsylvania, we can go back further for legality. So I would have them weigh in on how long you should be keeping that documentation. And let me look, I think there was one more question, Denise. Oh, requirements for the power of magnification. We're going to have to do a little research on that. I know I have been told in the past by people I consider to be real gurus of sterile processing that you needed to have extremely good lighting and at least 5-time magnification. But we're going to have to do a little more research on that. I don't want to give you an exact number without looking that up.
Thank you, Joanne. And one other thing I wanted to mention here, I'm going to put in the chat. It's something that has come up here recently about tracking instruments. I know Joanne was working with facilities on this. You don't want to use tape, right? That's an infection prevention no-no, and you could get cited on that. There are other ways to track instruments. You can have a fancy system, and those are all really good too. But you can also, in this article, you can use your chemical indicators within your packs and if you can write on them, you can then put it with the patient information. Or you could write it on an envelope and put it with the patient information, but that would help you. I know we're running out of time, but would help you track instruments to a patient. So something else important to consider for your facility.
Okay, as Denise said, we are at time. I want to thank everyone for joining us today. If you experienced any issues accessing the evaluation or certificate of continuing education, please feel free to direct any inquiries to Shelley Mixel at shmixell@pa.gov. This concludes our webinar.